All right, so now we need to talk about transporters and their role in terminating the synaptic signal and replenishing the pool of neurotransmitter. All right, so I want you to think about where and why neurons need transporters. And again, I want you to stop the lecture and write your own answer down so that you can do your own thinking. All right, I'm glad you're back. The answer is pretty simple. We certainly need transporters to remove neurotransmitter from the synaptic cleft in order to terminate transmission. The signal, the binding of transmitter to the receptor is transient, governed by mass action, but transmitter will continue to stimulate those receptors unless removed from the cleft. And we need to replenish locally synaptic vesicles store of neurotransmitter in order to make the recycling of the vesicles efficacious. And then generally, we need transporters in all cells, including neurons, to bring into the cell needed metabolites and substances, whether it's from oxygen and glucose to small peptides and amino acids, but for general metabolic needs. All right, so what would then be all of the different kinds of functions this process of transporting neurotransmitters might subserve? Again, take a break and make your own list. Welcome back. All right, so we've talked about terminating synaptic transmitter, but removing transmitter from the cleft may also regulate the length of a synaptic signal and how long that signal will be maintained. It certainly can be used to prevent the spread of neurotransmitters to nearby synapses. By removing transmitter from the cleft, there's less diffusion, so there'll be less crosstalk between different synapses. And a transporter can actually work in any direction. So if various gradients are favorable, we could actually use the transporters to release cytosolic neurotransmitter into the extracellular space. So they can operate in reverse and provide means for calcium-independent transmitter release. We talked about metabolic uptake for metabolites, but neurotransmitters sometimes overstimulate their postsynaptic receptors. So the presence of transporters, again, might be useful in preventing that toxicity. And then replenishing vesicles in the releasable pool. But we also have to think about this background level of neurotransmitter in the extracellular space as possibly defining a signal-to-noise ratio. So the background amount of neurotransmitter that's just running around the extracellular space might indicate just the random noise level for activating receptors postsynaptically. And when we get an evoked release, we want to be able to see a signal that's above that noise. So by removing transmitter from the cleft, transporters are actually increasing that signal-to-noise ratio. All right, what are the theoretical ways to get a substance across a lipid bilayer? Again, do your own thinking and pause for a moment. All right, welcome back. We have passive movement of substances down a concentration gradient or down an electrical gradient, which we've seen when channels open. You've already looked at voltage-dependent channels, or channels that might be open in some other ligand-gated way. But the equilibrium calculations from a Nernst kind of behavior certainly is the fastest way for substances, especially small ions, to get across a lipid bilayer. But for larger molecules, we have something we think about as being primary active transport. Used to be called pumps, but these are movements of the substance against their concentration gradient. And this is a relatively faster process. These kinds of movements can be coupled to energy in order to move up a concentration gradient. So we could utilize any high energy phosphate bond or redox energy or even light energy. And these larger molecules take advantage of that, and we used to call those carriers. Now everything is really a transporter. But these utilize hydrolysis of a chemical bond as an energy source, and they take a little longer. Then there's what's called secondary active transporter, an exchange or a co-transport or a counter-ion transport, where the substance itself is not moving down its own concentration gradient, but it's utilizing a secondary ion to move down the secondary ion's concentration gradient and moving the primary ion either up a concentration gradient or across a membrane, overall creating a negative free energy balance, so it's favorable due to the gradient on the secondary ion rather than the primary ion or metabolite that's moving. And lastly, we could have a substance bind to a receptor, and that bound receptor would then be internalized. And this is clearly the slowest process.